who are hearing what we're struggling with, in part, because I'll tell you, I've been at the LCB not quite 2 years as chair, and so the world of cannabis is still relatively new to me. And one of the things I learned very quickly when I got here in March of last year was while we were debating some new development and thought we had come to a consensus on how to approach, there was something even newer approaching that we had to also worry about. And that feeling of just sort of constant evolution and revolution has not slowed at all. And as I read the cannabis media around the country and around the world, all regulators are struggling with these same things. And I'm not an expert. Luckily, Dr. Hoffman and Justin Nordhorn and others inside the LCB are, but they also then need to go not only educate board members, but then go to the legislature and educate politicians on the fast moving science behind this. And that's one of the struggles that I think anybody who's in the community of I would say, if not supporting, at least recognizing and accepting legal, regulated recreational cannabis, um, is these decisions are made by people who know less than you all. And frankly, a lot of them are made by people who know less than I do, the people in our authorizing environment. And I think science, I'm hoping will be sort of a connective tissue for us, that when we go to the legislature, we get these sorts of questions about what does the science say. And if you have any experience up there at all, or in testifying in Congress,
Oh, not
science.
Congress, court.
Yeah,
I have
well,
testified in court before. I've been an expert witness. My field is forensics. So
uh, and
good morning, by the way.
good morning. And it's a different world when they sit across the way, and they might hear from somebody who's got years of experience and has done this, but the political lens looking at science is not all the same. So what I'm hoping is that this group, that you all can really lean on your expertise. Be in the charter it talks about working collaboratively and transparently. To me, those are the only requirements we should have. And if we do that, and we can get some healthy debate, and you can help us, keep up to date more on what's going on, and then help us with that next step, which is really our job. How do we take what's happening out there and continue it in a system where we're regulating for the public safety and the public health, as well as wanting a robust industry? We're not in the business of prohibition. We have possible, though I'm not optimistic about federal legalization coming. How would that change these things? Do we set up a system of our own that we have, 20 some legal states all with different, or is it time to think about what are the universal, if there are, kind of scientific standards for some of these things? I mean, it was hard enough for me to learn how to pronounce cannabinoids. So imagine when I'm hearing about, D8 and D10 and CBN and all these other things. It's a lot for us. So with that, I'll turn it back over to Dr. Hoffman, and I appreciate the work that she and her team did in bringing you all together. And also, one last note, just as an example of, I think, how excited we are about having a group like this coming together for the first time. We have 3 board members, and because we have 3, no 2 of us can be in a meeting together unless it's advertised public meeting, because that is a quorum, and so we have to separate. So I'm going to do the first 30 minutes of this meeting and listen in, and then member Jim Vollendroff, who some of you may know and has a long history in the world of addiction studies and prevention, is our newest board member, and he's going to join at half past the hour because he also has a great interest in this. So I will have to ring off at that point just to allow him to have that space. So with that, I'll just thank you all. And yes, is there a question, Chris?
No
Oh,
question.
oh, I just popped up there.
I had been disconnected.
Oh, okay, got it. Okay, great. Back to you, Kathy. Thank you all.
Thank you, Chair Postman. And I want to first, before we begin, just really extend our thanks to everyone for being here again, and also for navigating, getting together from all over the place in a virtual environment. We're going to have hiccups along the way. Dr. Beecher's already had that happen this morning where he got kicked off and now he's back in again. So happy to be gracious and flexible with everyone moving forward. Another thing I want to share with everyone is we are recording this meeting. We started recording when Chair Postman began speaking. A couple of reasons for that, so we can look back on this meeting in the future as we move forward. And so our initial meeting here will be recorded, and all of our meetings will be recorded as well. So just wanted to make sure that everyone was aware of that. So I'm gonna share my screen really briefly, go over our agenda really briefly, and then we can start with introductions, okay? All right, thanks for that. All right, so again, we've already, I think, sort of gone through a couple of these agenda items already. We are here at the 10:10 a.m. Part of the meeting. And we do hope today to introduce ourselves, share some ideas. So in some of your initial, my outreach to you asked each of you to come to this meeting today with one question that really interested you about cannabinoids. And then we'll talk a little bit about co-chair selection. I am the chair of this work group, but would like to have one of you join me in kind of leading our topics as we meet in the next year. And then we'll talk about next steps, our next meeting, talk about meeting schedule, because we do have 7 meetings scheduled into the future, and then hopefully adjourn on time at 11 a.m. So just briefly to give just some, and I'm just going to do the 1 minute, the nutshell version of how we got here. As you know, the topic of cannabinoids has been in the forefront of cannabis regulation from quite some time, and specifically in the last couple of years. In Washington State, we tried to move some legislation forward from various parts of the industry and within the government to kind of decide how we regulate cannabinoids, how we regulate THC. And we learned along the way that there's a lot we don't know about. Their Postman just shared. So we crafted this group sort of based off a piece of legislation that one of our senators introduced last year that didn't move in the last session. So that's kind of the construction. And I want to emphasize that the construction of this group was based on the word may. It may include this group of people, it could include others. And so that's why we cast such a broad net in bringing everyone together. And so we really appreciate the diversity of this group. And so with that, I'd like to hand it over to Justin Nordhorn, who's our Director of Outreach and Regulatory Affairs. I think I got that right, Justin, to see if there's any additional comments you'd like to add. And in the meantime, I'm going to go off camera and see if there's anybody else I've forgotten to admit, and then we'll get going. Thanks, everyone.
Thank you, Kathy. Well, welcome, everyone. I'll keep this brief so you all can get into the conversation. But again, as you've heard, we just want to thank you for your time volunteering to come to this work group. This is really important work for us as we look at cannabis. Obviously, it's been around for years and years and years, but there's so much more attention on it now, and a lot of that goes to the product, which goes to the science. And so where do we go to set up some of the policy aspects around this? So we've had a lot of interest in this. When we put the call out for applications, we had national and international interest in this. And so we really are going to have this, vast breadth of knowledge out here, and we're really excited about bringing all of that together so we can make some informed decisions. Just a quick thank you to Kathy for taking the lead on this, designing and leading this program, and getting this off the ground. This is a lot of work, and we're really excited on where it's going. So really appreciate everybody's time. And with that, I'll turn it back over to Kathy.
Great, thanks very much, Justin. So now let's do introductions. I know we had 14 people that we invited to join this work group and who accepted our invitation. We do have 2 that aren't with us today. One from Warsaw, Poland. I was really sad to hear that she was not able to join. She wasn't feeling well. So at the conclusion of our introductions, I'll share what she sent to me today to share with you. But I'll go ahead and kick it off. I'm Kathy Hoffman. I do have a PhD in leadership and change in complex regulatory systems with an emphasis on cannabis regulation. So that's kind of my background. I'll just start with the person I see in the corner of the screen, and that is Taylor Carter. Taylor, if you could give us like 2 minutes, a little bit about your background and the question that interests you most, and go ahead.
Sure. So I'm Dr. Taylor Carter. I'm a postdoc at the University of South Carolina School of Medicine, so other side of the country. I work specifically with cannabinoids' effect on the immune system. My specialty relies in bone marrow differentiation and the effect that cannabinoids have within that system. Our lab itself also focuses on many disease and inflammatory models, looking at cannabinoids' effect. We work with CBD, Delta 8, Delta 9. A lot of my work itself within the bone marrow focuses on a lot of the receptors that these cannabinoids bind to. Again, we all know CB1 and CB2, but I focus on things such as GPR18, GPR55, the TRP receptors, as well as some of the other receptors that the cannabinoids kind of affect. I do a lot of work with transcriptome, so looking at RNA-seq, microRNA-seq, single-cell RNA-seq, and I do a lot of flow cytometry. So I kind of do the whole immune side of research, but I focus specifically on cannabinoids. I've brought THC stuff to AAI, the American Association of Immunology conference last year in Portland. This year I'll be bringing THC data to SOT with Society of Toxicology, as well as AAI again. So trying to bring cannabinoids to immunology. So this is a little more cannabinoid focused, but the question that I probably have and what I'm kind of focusing on right now is the effect of cannabinoids on development, whether that be fetal development or childhood development, brain development. Again, I'm seeing it in the immune side. I'm just wondering how that kind of plays out, larger question based of age that cannabis should be, I guess, recommended for or allowed. So that was my introduction. Thank you.
Thank you very much. So moving on, Dr. Klein.
Thank you. I'm Tracy Klein. I'm a family nurse practitioner and associate professor at Washington State University. I also work along with Dr. Gang at the CCPRO, and he'll probably talk about that in just a moment. My interest really in cannabis came from both being a clinician and also being someone who teaches pharmacology. I work with PharmDs in their teaching residency as well. And in that work, we had a lot of questions come to us about cannabis and started doing our own research in order to be able to answer questions that clinicians and pharmacists were asking us. There's still, of course, a lot to do in that area, so I'm not sure I could pick any one question. But just suffice to say, I'm most interested in the therapeutic aspects of cannabis.
Thank you very much. Holly, please.
Hello, my name is Holly Moody, and I actually most of my experience with cannabis before probably 2020, since Arizona was the last state to implement testing for its medical marijuana, was in the forensic field. I did a lot of toxicology work after I got my master's degree in forensic science. I have testified in court to a lot of those values results that I've come up with through the years in toxicology. I'm trying to be brief. My interest in cannabis lies in the testing of it. Currently, I do the residual solvents testing here for compliance testing in Arizona, up to Arizona DHS limits. I have a lot of questions personally that lie in some of the analytes that they have us testing for and safety. They should just follow a USP method, but they don't. And they kind of implement their regulations on the fly. But my one question that I have is, and I'm looking at it because I wrote it down, was I'm wondering about the metabolites that they use to test for DUI. Because in Arizona, we have a unique situation, and that is the DUI law for cannabis says that the metabolite of cannabis, which comes out to be 11-hydroxy THC, is all they use because of the half-life of that. If you have a medical card in Arizona, you can have any other metabolite other than 11-hydroxy in your sample, and you won't get a DUI. It's unique in the country. I am glad to have the chance to join you all and put our heads together to come up with some solutions for the entire industry, potentially.
Great, thank you very much. Moving on, Dr. Sams.
Yes, thank you. Good morning. I apologize for my voice. I'm coming off a recent bout with COVID. I'm Dr. Richard Sams. I'm the scientific director of KCA Laboratories in Nicholasville, Kentucky. The laboratory's full service, ISO accredited testing laboratory that services the hemp and cannabis markets. Previously, I was a tenured faculty member at the Ohio State University College of Veterinary Medicine, where I taught veterinary pharmacology. I developed bioanalytical methods, performed pharmacokinetic and drug metabolism studies, and I was responsible for testing samples collected from racehorses for the presence of prohibited drugs. You can probably imagine that some of the reports issued by the laboratory were contentious and led to extensive legal challenges. At KCA Laboratories, we've become extensively involved in the analysis of semi-synthetic derivatives of cannabidiol that entered the market soon after passage of the 2018 Farm Bill. Initially, these substances were limited to Delta-8 THC, but more recently we've seen Delta-10 THC, Delta-6A, 10A THC, hexahydrocannabinol, and acetate derivatives of all of these substances. More recently, we are seeing products with altered alkyl side chains instead of the pentyl group that we're accustomed to seeing with THC and CBD. We've identified deficiencies in some of the standard methods that are used for determining cannabinoids in plant material, but these methods aren't sufficiently selective to differentiate the semi-synthetic materials from the phytocannabinoids as well as the synthetic byproducts from this reaction with CBD. In terms of the major question, I believe that we need to know more about the potential for drug-drug interactions between THC and prescription and over-the-counter medications. Thank you.
Thank you very much. All right, moving on, Dr. McClay.
Hello, everyone. My name is Bill McClay. I'm a pharmacist. I'm licensed in Pennsylvania and Ohio. I currently work as the lead medical professional for Ethos Cannabis. It's a multi-state organization. I manage a team of medical professionals in Ohio, Pennsylvania, Maryland, and Massachusetts. There's many different laws, different products to deal with across those states. We work as a collective to try and treat our patients accordingly. I've been doing this about just over 3 years, and I've seen thousands of patients in consultations over the past few years. Unbelievable success in treating multiple disease states. We try to collaborate with the patient's doctors, oncologists, surgeons. It's been unbelievable in Pennsylvania, especially. I think most people know it's been a pretty decent success over the last couple of years in the Pennsylvania market, working with the Department of Health and trying to grow this medical marijuana program. I'm always interested in research. I think this is a fantastic group to be able to pass around ideas with different backgrounds. Something that I've been thinking about lately, one question that came to mind, which is kind of a hot button topic in dispensary world right now, is cannabinol with THC and its effects on sleep. There's really no solid evidence out there for its effects, but anecdotally, we've seen really good effects for people, for pain, for insomnia, and I'd love to know why. Why are the studies so small, and why are the results so varied? And what can we do moving forward to try and utilize it the best we can?
Thank you very much. Sarah, Ms. Murray, please.
I'm Sarah Murray. I am a nurse in Walla Walla, Washington. Right now, I work specifically with substance use disorder folks, but I have some interest in this work group because I have worked with people over a couple of decades in the healthcare field, and the therapeutic applications, of course, of cannabinoids are vast, but also in the current world that I'm in, there's just not enough known about cannabinoids for recommendations for patients. And I do come from a therapeutic application, but I also would like to consider regulatory effects of cannabinoids. Like Oregon was developing a drunk driving test based on cannabinoid breathalyzing, and I just feel like the science for this is not there, and what they're analyzing is not there. And I am excited to come together with a large group of people with a great, vast knowledge of compounds and toxicology and chemistry, so that we can put it all together and actually come up with something that means something, and having some guidelines for like what you're measuring. Is it in micrograms per milliliter? What are we doing with this, and what does it actually mean, and what are those percentages on people's baggies? Like, I know what works for me, but does that actually mean anything for somebody with glaucoma, and how do we tie that all together?
All right, thank you very much for that. So let's move on to folks who are not on camera, and I would like to invite you to come on camera when you introduce yourself, but let's start with Amber Wise.
Hi, good morning. I am Amber Wise. I'm the science director at Medicine Creek Analytics, which is a testing lab here in Washington State. We are tribally owned by the Puyallup Native American tribe. I have a PhD in chemistry. I was previously a faculty member, chemistry professor, and I have previously worked for an extraction company, so I have a little bit of understanding of some of the motives of producer processors. And I've been managing this lab for 4 years now, and we've done some peer review publications. And I'm hoping to set up another couple of research projects in the coming year as well. So really trying to professionally move the science forward in the cannabis space as much as I'm able to in my capacity. I have many questions that I'd like to know about cannabinoids, but I think fundamentally, super simple and easy. How does the endocannabinoid system really work? And, I think that touches on almost everyone's questions here fundamentally. And how do we really start to integrate this into medical educational programs? We know it's real, we know it's a thing. How do we get that information out there, to the folks who need to understand it as well? So I'll leave it there for now. Thanks for having me.
Thanks very much. And Dr. Gang, since you came on camera, go ahead.
Kathy, can I just jump in for 1 second? Sorry to
Of
interrupt.
course. Yes.
I'm just going to say my goodbyes and introduce you to board member Jim Vollendroff, who has joined us. Maybe at the end of these introductions, he can just have a quick minute to give you a little more on his background. But thank you all again, and enjoy it, Jim. See you later.
Thank you, David. Thank you.
All right, should I go then?
Yes, I think something froze on my end. So if my screen was frozen, it was because I just was getting this little error message. So please go ahead.
Okay, you're back. Now you're back, Kathy. Great. Yeah, I'm David Gang. I'm a professor in the Institute of Biological Chemistry at Washington State University. I'm also the director of WSU's Center for Cannabis Policy Research and Outreach, which Tracy Klein is our assistant director, and she mentioned it earlier. We also call it CC Pro. for short. I've been involved in studying medicinal plants for going on 30 years now, I think. It's hard to believe how time flies. I was part of a group that published the first paper looking at how cannabinoids might be produced in the trichomes, looking at a genome scale kind of a study. The first one of those was published over a dozen years ago. We haven't been able to do much work on the plant since then because of regulatory issues. That's kind of changed because hemp legalization. So I'm excited to be able to do a lot more in my area, but I'm a plant biochemist by training, and I'm very interested in how plants like cannabis sativa make the interesting compounds that they do and what they're useful for. And that's obviously, from what people have already said today, it's clear it's useful for many interesting things. I was thinking about the question that you asked. Like Amber, I have a whole bunch of questions actually. The one that came to my mind related to this group was how do we think about regulating the components of this plant with how fast things are changing with regards to the science, and how do we understand how best to manage it in that kind of an environment, so that number one, we're able to protect the safety of the public, and number two, also help support an industry that is, for our state anyway, significant economic benefit. But how do we merge all of those interests in a way that, for the greater society good is going to be a positive thing? And I think it's a challenging question, but I think it's something that the science is going to be playing a critical role in being able to come up to answers to that question. So I'm really excited to see that this group has been convened so that we can get people from all these different perspectives together to talk about these questions in a way that I think hasn't been able to be done before. So I'm looking forward to seeing this move forward.
Great, and thanks for joining us. Just to recognize there are some people on this group who've helped us in our deliberative dialogues sort of leading up to where we are today in Washington State, at least, and are thinking about regulating these cannabinoids. And Dr. Gang was in that part of that original group, I think it's fair to say, right? Right. All right, so moving on to folks who are not on camera, let's move to Brad Douglas. I think your camera and your mic are both enabled on our end. If you're not able to go, just go ahead and maybe we'll come back to you, Brad, when we have a minute. To that end, let's go ahead and move on to Jessica Tinani. And Jessica, you are also, your mic and your camera are both abled from our end. Okay, go ahead, Amber.
Sorry, I just got a text both from Jessica and Brad that neither one of them can unmute.
They cannot unmute. Okay, so let me, okay, well, shoot, we're gonna have, I am gonna allow Jessica's mic again and allow Brad's mic again. But I've done everything I can on this end to enable the microphones for them. Shoot, I guess we'll have to come back to them. So we'll do a follow-up with Brad and Jessica at the next meeting, unless Brad and Jessica, you'd like to send me an email and I can share it with everyone really quickly, because I do have an email. Go ahead, Chris.
When it happened to me, I unattached and rejoined, and it worked again. But
it worked for you. Okay.
So
Holly's raising a thumb, so
they could do that quick.
Okay. Let's see. Let me see if I can find Brad here again. Oh, it looks like they both jumped off here. Okay, so we'll give them a minute to try to rejoin, but in the meantime, I will share our screen with a message from Dr. Binkowska. She's from Warsaw, and provided this message to us today, and I'll read it for folks who may be joining us on the phone. Dr. Binkowska is a psychologist with a PhD in cognitive neuroscience. Her research interest and work focus on the effects of cannabis on the human body and behavior. She's head of drug teams at Neurocognitive Research Center at SWPS University in Warsaw, and head of R&D at PredictWatch Company, where she's working on developing evidence-based mobile solutions dedicated to harm reduction and problematic substance use. And then her question, and it's kind of a lengthy one, but I think it's something we've heard some of you say this morning, and that is, for me, a really important research question with strong practical implications. That is, in the case of DUI with cannabis, is how to assess that people are experiencing acute cannabis effects, intoxication, both in the case of recreational and medical cannabis use, recreational and cannabis use. Based on research so far, we know that blood THC concentration is not an accurate indicator of intoxication level and cognitive psychomotor impairment. It's essential to develop other biological and behavioral ways to assess functional impairment due to THC intoxication, taking into account if it's medical or recreational use. All right, so I will stop sharing my screen for a moment and see if we can bring Brad and Jessica back on. I don't see Brad. And here's Jessica. So Jessica's still here.
I think I got back on, Kathy. I think
You're
am
on.
I
We can hear you.
Excellent. I got off and got back on. So that
Okay.
Thank you, whoever suggested that.
Yes.
So my name is Jessica Tanani. I am the CEO of VertiBio Research. We got issued the first cannabis research license in the country by the Liquor Cannabis Board several years ago. My background's really traditional biotech. So I think we've kind of looked at the plant fairly traditionally. We were one of the first to release a public genome of the cannabis plant. We've also collaborated with groups like Argonne National Labs on the microbiome project of the plant, so knowing what bacteria naturally occur on the plant. We've also worked with other universities, so I think we're kind of one of the first groups to bridge that gap between working with a Schedule I plant and working with universities. I think my biggest question is, how do you kind of look at regulating? I think David kind of approached this diverse set of compounds with very different risk reward around them. You know, there's very different effects that these compounds have, and you can't really bucket them all together. So, you know, how do we set up a system that we regulate things that, you know, have the potential of getting people high, and compounds that don't, even though they fall into the same bucket, and kind of look at that regulation and do it soundly.
Great. Thank you very much, Jessica. I'm still looking for Brad. It looks like. Yeah, how unfortunate. We'll have to have Brad give us an intro, or wait for an email, or we'll send something out afterwards so everyone can get a bit of Brad's background and his research interest here, unless he's texted someone, Jessica or Amber. I know that happens sometimes in our meetings. All right, so anything to add, Jessica? It looked like you were unmuted there for a minute.
No, he just said he tried to get back on and off and it didn't work, but if there's any update, I'll let you know.
Okay, sounds great. I want to pause here really briefly, and Board Member Vollendorf, did you get an opportunity to introduce yourself when I kind of went offline for a minute and wasn't in the conversation?
I did not. May I take a moment?
Okay, so please do. Yes.
Great.
Please.
Hey, thank you so much. And thanks everybody for participating. I'm super excited about this science work group. I want to acknowledge Kathy Hoffman for her work in helping pull this together. I can't think of a more qualified person within the LCB to do this, and want to thank you all for your time. Just by way of background quickly, I most recently come from the University of Washington, where I helped develop the Behavioral Health Institute Institute at Harborview Medical Center after working for King County as the director for their behavioral health system here in Washington State. And so I am really interested, particularly having just recently come from the University of Washington. I still teach in the School of Social Work at the University of Washington, so I still keep affiliation with the university. But I really am interested in research and how can we use good, solid research to implement evidence-based policymaking. And so I'm really excited about this work and future research opportunities. I'm looking forward to hearing everybody's response to the question about a research topic that interests you, because there are, I mean, I could go on and on about research topics that interest me related to this, so I'm super excited to hear from all of you. I do need to jump off at 5 minutes before 11, so I won't be on for the entire time, but I do plan on joining in the future. And one last thing I'll say is I'm also a person with lived experience, meaning I'm in recovery from substance use disorder, and I bring that background and experience to the conversation and table as well. I think that's an important voice that needs to be a part of the conversation, so I offer that as well. And again, want to just thank you for your commitment to this, and look forward to seeing the amazing things that come from the convening of this group. So thank you.
Fantastic. Thank you, Board Member Vollendroff. We do have one more work group member who has joined us, and that is Ryan McLaughlin. Ryan, you should be able to come on camera, and you should
Great.
be able to
Can you see me
enable your
now?
mic.
Can
I'm
you
sorry?
hear me?
There you are.
Okay,
Thank you.
I'm new to Microsoft Teams, so I have no idea if this is working. Hello, everyone. It's nice to meet you all. My name is Ryan McLaughlin. I'm an associate professor in the Department of Integrative Physiology and Neuroscience at Washington State University. My lab really focuses on using preclinical animal models to understand both acute and long-term effects of cannabis, mostly on brain and behavior. More specifically, my research questions really are inclined towards understanding long-term developmental effects of cannabis. So we've developed a novel approach using rats and mice where we can allow rats to self-administer whole plant cannabis extracts. So they make responses for brief puffs of cannabis vapor that come into the chamber. And we can look at motivational properties of cannabis, look at brain mechanisms that drive problematic cannabis-seeking behaviors in rodents, and look at the long-term ramifications of those, like I said, mostly in the context of developmental exposure, i.e. Cannabis use during pregnancy. So for my most interesting sort of follow-up questions and things we need to really be digging into are really focusing on this in terms of what are the long-term effects of developmental cannabis exposure, not just for developing offspring, but also use of cannabis during pregnancy for the mother, and how cannabis use during pregnancy can interact with hormones to influence susceptibility for postpartum depressive-like symptoms and things. I think that's a wide open area of questioning that we can start to look into. So yeah, that's me in a nutshell. I'm really excited and I'm really honored to be part of this group.
Well, thank you very much for joining us, and apologies for learning yet another new virtual platform. It seems like there have been so many in the last 2.5 years, so we're still working on. I will say here at LCB, we're still working on figuring out Teams. It just changes all the time. So appreciates everyone's graciousness and patience moving forward in this space. So I think we've touched on everyone. Go ahead, Jessica. Do you have an update?
I was going to say, Brad says he's on.
Okay.
That he's on.
Let's see if we can find him here. Here he is. And so Brad, your mic and your camera should both be on.
I am here, I believe.
Oh, yay.
Well, I just figured out that I can reinstall Teams in about 4 minutes, so it's good to have that benchmark. I'm Dr. Brad Douglas. My academic background is in organic and medicinal chemistry with some work in targeting drugs to the brain. In the private sector in cannabis, I've been involved with a number of different companies, some plant-touching THC cannabis, some on the hemp realm of things. I've also spent some time as a lab director at a Washington State cannabis lab. Professionally, I also have a decade and a half experience with FDA affairs, mostly foods and dietary supplements, but also some drugs. As far as a research question, there's many, like many others here. I think one that I'm particularly interested in is the natural abundance of less common stereoisomers of THC molecules in particular, and their different pharmacology, whether that's physiological effects that we can harness or toxicological effects that we might want to be aware of. But other than that, it's just great to be here with all of you. Looking forward to answering some fun questions. And I will note that although this meeting's about to end, I'm in the midst of a snow date here with small children. So if there's banging in the background, you'll forgive me.
It's our pleasure, Brad, and also for those of us who have pets who respond to doorbells, doors knocking, things falling. Yes, we're all there with you. So I guess that's the reality of the virtual environment, right? Life keeps happening even though we're on camera. So happy to work with you on that. So I think that's everyone. I did want to mention Jillian Shower.
I did.
Oh, go ahead,
I
Chris.
would still love to. So the same thing. I mean, I'm actually going to echo so many of you. My background is natural products chemist, taught through the system at the University of Illinois, then University of Michigan, University of Illinois College of Pharmacy, University of Michigan Medical School, and then University of Florida. Switching from a high throughput qualitative analytical drug discovery program to a quantitative series of techniques. About 3 years ago, we began working up a quantitative analysis of cannabinoids because I live in North Carolina and it's not yet legal here. We had to do the work in Holland, which was quite successful, but developing a series of techniques. So fundamentally a natural products chemist, I've known the chemistry of cannabis for ages. The question is the same to me. The most important question, I mean, there are the biological questions, but the products are right now so poorly labeled when it comes to quantitation. I really think we need something simple like the concept of proof in alcohol or in cannabinoids. You need some way of cutting through the psychologically active and the non-psychologically active and creating some sort of a simple scale that tells you what is actually in the end product. It is ridiculous to see something that says I'm 99% THC, less than 1% CBD, without any concept as to what the percentage of that is in the entire material. So every product actually has to be better labeled. What I see in the market now is just awful. But the biology. My wife is in terrible pain, and so CBD is a great help. But my understanding is, and THC CBD is a better combination for topical use. And I mean, these things, the products need to be developed. There's a huge market out there. Jessica.
Thank you very much for that. Okay, so I think everyone's had an opportunity to weigh in, provide their background, and provide at least one of the many research questions that you have been carrying around for the last several years. Did I get that right? I hope I did. Okay. Fantastic. And again, Jillian Shower wasn't able to join us here today. She had a conflict. She will be joining us in future meetings. And she'll be able to provide her background when she joins us as well. So we are running a little bit behind schedule. I don't want to spend a lot of time, and I don't want us to run overtime either. I don't want to spend a lot of time trying to catch up, but I would like to spend just a few minutes looking at the draft charter. I want to share that we kept the charter pretty general to give this group the kind of flexibility that we thought was important for these discussions, because as I think we've just noticed, people have a lot of they are differing research questions that can take us down a lot of different roads, I think. So, and we also wanted to make sure that we had the flexibility to create subgroups if people were interested in that. So again, the purpose of this group is to provide a forum and platform for scientific and data-driven discussion about cannabinoids, and there are things that we may do. The other thing I wanted to say is that we're all here together to talk about the issues that we'll identify as important to this group in the next meeting, now that we've all gotten together and shared our thoughts. But we may also bring other people in, and I just wanted to leave that open as well within the charter to say, even in these subgroups, we may bring other experts in to help inform our pathway forward and the breadth and depth of our discussion. So we didn't want to kind of close off that resource in the future. Again, with respect to meeting, once every other month. We know that you're all terribly busy. We also know that you're volunteering to help us in this really important work. So we wanted to be thoughtful about how we scheduled those meetings. So at this point, we have a meeting in February, April, June, August, October, and December. And that does give time for subgroups if we want to have them at some point in the future during this work. And those are the ad hoc meetings. And then with respect to communications and reporting, there's a lot of material that's going to come from this initial meeting, I can already tell. And so I will work with our support staff at LCB to put notes together on that, including a detailed list of all of the interests that you brought to the table today. And we'll share those out with you as a group, and then also on our outward facing webpage. If you missed the email, or if anybody who's listening in wants to follow up and see what we've been doing and how and why and all those sorts of things. And we'll also have minutes that we'll go over at each meeting as well, just to make sure that we captured everything correctly. We'll share those before we meet so you can review them. So my vision for that is providing you with a meeting packet at least 2 weeks before the meeting occurs. That okay with everybody? Is that enough time for review? Okay. All right, so in the interest of time, if you do have any additional things that you think we should include in the charter, please don't hesitate to reach out, and we'll bring it to the next meeting if that's okay. All right, so I'm going to stop sharing the screen. Next item on the agenda was choosing a co-chair, because I think this would be really fun to do it by myself, but I also think it would be great to have somebody co-chair with me. So wondering if anybody wants to just jump in there and volunteer, or how we might want to go about selecting a co-chair. Jessica, you're smiling and laughing.
Laughing's not volunteering, Kathy. A couple of us just got off a similar committee and may have a little bit of
PTSD. Yes, gotcha.
But
gotcha.
If nobody else volunteers, I'd be willing to help you, but there may potentially be a better person in the group.
Sure. So I will say, Jessica, there are several of you that I've worked with in the past. Happy to work with you again, but if there's someone who I haven't worked with in the past who'd like to raise their hand, happy to hear from you.
I would do it. I'd be more than happy to.
And who is that? You, Holly?
Yes.
Okay. Well, that sounds great.
Just let me know what I need to do. Follow up with me in the email. We can start doing that whenever you're ready.
Okay, fantastic. So thanks for that, Holly. So we'll go. If any objections from anyone? Okay, great. So Holly, you are the co-chair. Thank you very much for volunteering. All right, so the final piece of this was really to just wrap up and talk about next steps. And I've kind of done that, I think, in a way. But I'd like to invite Chair Vollendroff to come back on camera and Justin Nordhorn to come back on camera and offer any closing remarks before we close out today. And then we'll look forward to our next meeting on February 1st. So Justin or I'm sorry, Board Member Vollendroff, any closing remarks before we sign off today?
So I'll just jump in really quick. So thank you again, everybody. It looks like from the introductions and the points of interest, and everybody came with a really solid point of interest. So I think this is going to be a really robust conversation over the next year. So really appreciate the thought that's been put into this so far. Thank you for the volunteers to be co-chair. Appreciate that as well. So we're looking forward to this experience.
Great, thank you, Justin. And I think Chair, I'm sorry, I keep calling him Chair. Board Member Vollendroff, I think had to leave at 5 minutes to 11, if I'm not mistaken. So that's why we're not hearing from him. All right, any closing remarks from anyone? And we'll do something really odd and conclude a meeting a few minutes early so you'll have time to get to your next meeting. Anything to share before we sign off? All right, everyone. Thanks again, as Justin noted, for your time. We really appreciate it. Looking forward to great discussions in the future. Look for minutes and a recording on our outward-facing webpage and links to your email box in the coming weeks. In the meantime, have a great holiday season. Take good care. See you next year. Thanks all.
Thank you.
Thanks,
Have a nice one. Thank you.